Hello, welcome everyone. Today it's my great pleasure to have Hector here with us. He did his bachelor at the UB here in Barcelona, then his master also in Barcelona in Valdebron, and his PhD finally at the UBF in Barcelona and the PRBB. There he was really interested in figuring out how innate immune responses work, and he wanted to develop this further during his postdoc and also related to hematopoietic stem cell function. That's why he became a postdoc in the lab of Dominic Bonnet in the Francis Crick Institute in London. And he was awarded a Key Candle Fellowship and also acquired now funding from the ESH, the European School of Hematology, and the SIA UK. And today he will give us a talk about the opposing effects of clonal hematopoiesis in stemness and immunity. So let us welcome Hector.
Thanks, René, for the introduction, and thanks, Marcus, for the invitation to be here. It's my first time in the Josep Carreras. It's a very nice institute and environment, so I'm really happy to see some familiar faces as well and meet you in person. I will present my postdoc work with Dominic Bonnet at the Francis Crick Institute. As René was saying, I take the chance also to encourage PhD students in the audience to apply for a postdoc position. In particular, is what you see today is of interest to you. I think Dominic will be also keen to receive applications for a postdoc there. So yeah, just to encourage you to go to the Crick. You will miss the sun at some point, but anyway, it's a fantastic place to do science. So when I arrived to the Crick Institute, I was initially intrigued by the idea that natural selection can occur not only at the level of a species, but also at the level of our tissues. And because actually the reality is that our tissues are not composed by genetically identical cell populations, but rather we accumulate mutant clones as we age and cells divide. And this is pretty normal. We all have mutant clones in our tissues. And the idea is that these mutant clones can actually lead to some tissue-specific pathologies, or if these mutant clones are somehow selected and start to expand, accumulate further mutations, they can lead to cancer. So these ideas is where I found clonal hematopoiesis really interested. Of course, it affects the blood. And as you know, we acquire these somatic mutations at the level of hematopoietic stem cells, and these mutations are transmitted into the immune progeny. And this is also the idea that you will see has influenced my postdoc work, because I found interesting the notion that how these mutant immune cells can actually shape the functionality of other tissues as they travel and infiltrate practically all our tissues. I don't think I need to introduce much this slide to you. It's just to remind you that clonal hematopoiesis is this initial step of pre-leukemia. Sometimes we refer clonal hematopoiesis as pre-leukemia. The individuals at this point are clinically healthy. And the important point for me is also interesting to target clonal hematopoiesis because it's a way to prevent the apparition of hematological malignancies before these symptoms appear, right? So, as you may know already, clonal hematopoiesis, as we were saying, is when this somatic mutation is acquired at the level of the hematopoietic stem cells. These mutant clones start to expand. They give rise to mutant immune progeny, and it usually affects, at this stage, one of these two genes, the NMT3A or TET2. As you know, epigenetic factors involved in the methylation and demethylation of DNA. This will also become relevant in my talk. And just three main characteristics that has also influenced my research questions is that mutations actually now we know that can occur pretty early on life. Both TET2 and DNMT3A mutations, for example, by 20 years of age, we can already have these mutations in our system, and then they start expanding at a certain growth rate until they become really prevalent as we are like 50 years of age or above. So with this idea in mind, I set up three different research questions. The first one is, to understand how this clonal selection occurs, which are the environmental conditions that can further accelerate the expansion of these TET2 or DNMT3A mutant clones. Then, related with this question, we envision a bit of ambitious, perhaps, question, which is trying to characterize environmental conditions that could actually select for non-malignant clones that can help to outcompete the pre-malignant clones that already expand with age. And finally, the third branch of my postdoc has been focused on the immune system and the possible contribution of the mutant immune system into the development of not only the progression of hematological malignancies, but as I was saying before, how this mutant immune system can influence pathologies in other tissues that have been actually associated with individuals with clonal hematopoiesis, such as severe infections or cardiovascular disease, pulmonary inflammatory disease. So the contribution of this mutant immune system into these pathologies. So I will start with this last project, which was recently published, and then I will show some preliminary and unpublished data. Before going into the results, just to show you that we, at the Dominic's lab, we address these questions at the human cell level. We isolate human hematopoietic stem cells from bone marrow or cord blood, and we introduce these TET2 or DNMT3A mutations by CRISPR, and then we have our mutant or wild-type stem cell to inject into immunodeficient mice, recreate a humanized hematopoietic system, and then we can study clonal hematopoiesis not only in the bone marrow, but in different tissues. And we can do so with a classic wild-type versus knockout comparison, or trying to model clonal hematopoiesis scenario by injecting just a fraction of mutant stem cells into these mice. So I will start showing you how it looks, the immune system in the bone marrow, the immune system that is reconstituted from mutant hematopoietic stem cells. The focusing on TET2 in this project, as you can see here, the immune repertoire of the TET2 mutant immune system is significantly different for some immune subsets. We identify that some of these immune subsets are involved in the neutrophil development. So that's why for this particular project, we focus on the neutrophil development, and for other immune subsets, we start also developing other projects, but today, focusing specifically on this, the impact of TET2 mutations in neutrophil development. We identify the 5 different subsets, from more neutrophil progenitor to more the differentiated neutrophil, and then how these subsets are represented in different tissues, and then the impact of the TET2 mutation into this neutrophil development. And as you can see here, in all the tissues, we found the same trend, which is the mutation when we have a TET2 mutant hematopoietic stem cell. It gives rise to a complete different route of neutrophil development. We have less neutrophil progenitors in the bone marrow or in the lung, but we have an expansion of differentiated neutrophils from TET2 mutant stem cells. Then we went into a pretty complete characterization of the phenotype of both neutrophil progenitors and differentiated neutrophils, and the characteristics of the TET2 mutation in these both cell types. Just to highlight some of these phenotypes, as I said, is in the paper. I'm happy to be contacted for any questions, but just to highlight the TET2 mutant neutrophils, the differentiated ones, are characterized by this low granule complexity. They also have more abundance of primary granules rather than secondary or tertiary granules, and we are trying now to apply this phenotype to see if we are able to identify individuals with CHIP without the need of genome sequencing. We have actually validated some of this data in patients, in individuals with TET2 mutations. And then also, I think for this audience is of interest that the transcriptome and epigenome of both differentiated neutrophils and neutrophil progenitors in both subsets, the transcriptome and epigenome was associated to immature stages. And finally, regarding the functionality, at the level of the neutrophil progenitors, as it is being described for the more stem cell compartment, the mutation is associated with an increase potential and increase self-renewal and output potential. So I will focus now on the functionality characterization of differentiated mature neutrophils. We first wanted to characterize the phagocytosis and inflammatory response. As you can see here, we show that mutant neutrophils have a reduced phagocytic capacity, and we have validated the implications of this reduced phagocytic capacity in vivo. We have also seen that once we have these humanized mice and we perform intranasal challenges of LPS, mutant neutrophils tend to have an exacerbated recruitment to sites of inflammation. And once they have infiltrated the tissue, they deploy a more exacerbated inflammatory response, which is consistent with some mouse work reporting that the mutation in TET2 is associated with this hyperinflammatory response in monocytes. So we kind of see similar things in human neutrophils. And then the third interesting immune function of neutrophils that we wanted to analyze was netosis. In particular, why we thought that netosis might be relevant? Because first of all, how netosis, well, first for the audience, netosis is this release of neutrophil extracellular traps are called just these nets of decondensed chromatin with antimicrobial proteins. And it was this critical step in the process of chromatin decondensation that make us think that the TET2 mutation could cause an impact due to the role on the demethylation of DNA. So it's reported that the mutation of TET2 is associated to a more methylated DNA that tend to affect the chromatin conformation. And I hope to receive some of the feedback from the audience, as I know that you are quite experts on this. The fact is that when the pathogen is too big to be phagocytotic, the neutrophil undergo this process of netosis. And as you can see here, this is how it looks by electron microscopy. You can see these nets, these traps of DNA released by the neutrophils. And what we first observed is that TET2 mutant neutrophils are able to also secrete these nets. But as we start seeing more and more pictures, and we also develop other methods to really quantify this net, we observe that there is some sort of difference here, and here we were able to quantify the area covered by these nets. And as you can see, or maybe appreciate from this picture, yes, mutant neutrophils can undergo mitosis, but the net that they release is different. It actually covers a smaller area. As you can see here, we have quantified the area of the net covered by the mutant neutrophil. It's smaller. And we have done some analysis at the epigenetic level, and we believe that is because the chromatin is also more compact, is more closed. It cannot be expanded as it is in the wild-type neutrophils. So we thought, what are the implications, the possible implications of these particular nets released by TET2 mutant neutrophils? And we thought about the implications that nets have in cardiovascular disease, for example. It has been actually reported quite nicely how when neutrophils are activated, either by encountering a pathogen or by tissue damage, they can be activated and they release these nets, which actually activate platelets and trigger the formation of thrombus. So there is this clear established link between nets and cardiovascular disease. And we know, we have seen before, that there is also the link between individuals with clonal hematopoiesis and higher risk to develop cardiovascular diseases. And it has been mainly explained by the inflammatory phenotype of monocytes and macrophages. Now, we ask if this link between TET2 mutations and nets can also be a new marker, a new molecular explanation for the link between individuals with TET2 mutations and the high risk of cardiovascular disease. Because it is important that neutrophils release these nets, but it's also critical that these nets are quickly degraded to avoid tissue damage and the formation of these clots. That's why we perform this analysis to quantify the time of net degradation. As you can see here, this is the signal of the net on time. And as we add human plasma into the system, the DNases that are present in the human plasma will quickly degrade the net. So the signal goes down quickly. Now, when we compare in the presence of human plasma the signal of the net produced by mutant neutrophils, what we observe is that it significantly stays longer in time in our system. That's why nets produced by these TET2 mutant neutrophils are more stable, are more difficult to dissolve. And we think this is an interesting new molecular candidate to explain the connection between cardiovascular disease or pulmonary inflammatory disease, where nets are also a cause of these diseases, and precisely the association with TET2-derived clonal hematopoiesis. I will just wrap up this. As I said, this is in the cell stem cell paper from last year, so happy to address any question regarding this project. I'm gonna now move to the second branch of the project, which is more at the level of the stem cells, as I said before, which are these environmental conditions that favor the expansion of TET2 mutant clones. For that, we have also performed different in vitro experiments following the same strategy as before, but instead of recreating the humanized mice model, we can manipulate these stem cells in vitro. And for example, one of the first experiments that we have done is this competition assay. We put in competition wild type and TET2 mutant stem cells, and then over a long period of time, we apply to the system different immune challenges to recreate different types of infections. As it has been reported in mouse models that infections favor TET2 mutant clones, we wanted to see the same at the human cell level. First in vitro, what we observe is that specifically upon LPS, among these immunological challenges, is where we observe a significant outgrowth of TET2-derived clonal hematopoiesis. So we decide to really validate and perform different types of kinetic assays with LPS. As you can see here, it expands the ratio of TET2 mutant cells expand over time. So to start understanding a bit of the molecular explanation of this TET2 selection, we perform bulk RNA-seq at day 4, where we basically have CD34 positive cells, HSPCs, and at week 2, when we already have some differentiated monocytes. So we have the stem and progenitor compartment on one side and the monocyte progeny on the other side, and analyzing in both cell compartments the impact of the mutation with regards to the response to LPS. And the first thing to say is that, as expected in both cell types, in wild type and in TET2 mutant cells, we observe an upregulation of the inflammatory response as a result of the LPS challenge. The point here was to compare how was this response. And when we do such comparison at the level of stem cells, comparing the mutant one versus the wild type one, we observe that the level of upregulation of the inflammatory response, TNF signaling, was downregulated in TET2 mutant stem cells. While on the other side, at the level of the monocyte progeny, we observe completely the opposite, that the inflammatory response was further exacerbated at the monocyte level. So we have this opposite impact of TET2 mutation with regards to the LPS response. Less inflammation in the stem cell compartment, more inflammation at the level of monocytes. And it was just recently that these results make me think about something that we include in the previous project. In the paper, we had, as I said before, this characterization of the different steps. We have the neutrophil precursor, the immature neutrophil, the mature neutrophil, and as I said before, both transcriptome and epigenome analysis of these three populations show that in each step there is a shift towards a more immature stage associated with the mutation. And with regards to the inflammatory response, as you can see here, we have here the neutrophil precursor and the immature and mature neutrophil. There is a shift. The mutation at the level of the neutrophil precursor is downregulated, associated to the TET2 mutation, but at some point in the trajectory of differentiation, the mutation now switches and is associated to a more exacerbated inflammatory phenotype, which is something that we are still trying to understand how this switch and this opposite impact can occur during the differentiation of neutrophils in this case. So going back to the project at the stem cell level, we also did some in vivo experiments. As you can see here, we inject in a 1-to-1 ratio TET2 mutant and wild type stem cells. We allow the system to be reconstituted in the mice. And as you can see here, we can track over time how the TET2-derived clonal hematopoiesis dynamic is. And in the absence of any further stimuli, what you can see is we lose TET2-derived clonal hematopoiesis. And actually, for example, even if we perform secondary transplant, we still don't see an advantage associated to TET2 mutation. And I have to say that this is contradictory with work done in mouse models. Now, we believe that these contradictory results might be because of specifics of experimental models. With mouse work, you have irradiation sometimes, you have preconditioning of the bone marrow niche. So the disruption of bone marrow niche can actually be a factor that selects for the TET2 mutant clone in these mouse experiments. In our case, there is no bone marrow niche preconditioning, there is no irradiation, so the human hematopoietic stem cells that we inject, they encounter a more naive bone marrow environment, and maybe that's why the TET2 mutant cells, they don't show any advantage because there is no further stress in the environment. Because when we apply LPS, several injections over different every other week, we then see the expansion of the TET2-derived clonal hematopoiesis very clear. So we perform single-cell RNA sequencing from these humanized mice, both at the level of the stem and progenitor compartment and at the level of the immune progeny. At the level of the stem progenitor compartment, we obtain this HUMAP, and we confidently characterize this subset as the one that retain a clear stem cell signature. I know that there are some papers from John Dick and Stephanie Shi that report this immune memory HSE after an immunological stress similar to ours. In our case, we were not able to confidently characterize this subset, maybe because also slight difference in the regimen of LPS treatment. It's true that in our case, we were focused on understanding the response in the acute phase. That's why this analysis is done just after 24 hours of the last LPS injection. Where in their work, I think they have waited until the stress was gone to see the memory in the stem cells. So in our case, when we analyze specifically in this stem cell subset, the impact of the LPS response and the inflammation in these mice, we observe that wild type stem cells clearly upregulate many activation markers, while at the level of the mutation and at the level of this specific stem cell cluster, such upregulation is clearly reduced. And for example, we have done also pathway analysis, and it's very interesting to see how, when we compare the conditions LPS versus untreated mice, the TET2 wild type stem cells clearly activate transcriptional pathways associated with the inflammatory response or cell cycle, while there is no significant activation in TET2 mutant stem cells. And the nice thing is that you need to remember that both wild type and mutant cells are present in the same mouse, in the same bone marrow. So they are both together, as it happened in individuals with clonal hematopoiesis. Now, at the level of the immune progeny, what we found first is the reported myeloid bias associated with TET2. The progeny from TET2 mutant stem cells is clearly myeloid bias. But the interesting thing that allowed the single cell is to see that within this myeloid bias, there are specific shifts in the heterogeneity of monocytes and macrophages. And we use this reference from Florent Ginoux, that I think some of you might be familiar, to really characterize the profile of the monocyte macrophage derived from the TET2 mutant stem cells. And this is basically the conserved monocyte macrophage signatures across human tissues. And we map into this reference data set the progeny from wild-type or TET2 mutant stem cells. And as you can see, they occupy different spaces into this hue map. And actually what it means is that the progeny from TET2 mutant stem cells is characterized by this clear expansion of hyperactivated monocytes, but also now we know that they also have interesting difference in the trajectories of macrophages. While in the wild-type progeny, we observe some macrophages that are associated with anti-inflammatory phenotype. Such macrophages are gone in the progeny from TET2 mutant stem cells. And this is just another way of representing in our data set the different signatures. And as you can see, while the ones associated with the TET2 mutation are hyperinflammatory or inflammatory subsets, the ones associated with wild-type progeny are actually anti-inflammatory macrophages. So to summarize a bit of all of these projects involving the TET2 mutation at the level of stem cells, we observe, first of all, this distinct response to different types of immune stress. So not all the immune stresses are going to push and favor TET2 mutant clones. In our case, it was specifically LPS, both chronic and acute challenges that caused the expansion of TET2-derived clonal hematopoiesis. And particularly at the level of the stem cells, we show transcriptional and functional refractoriness to this inflammatory stress. And at the level of the immune progeny, as we saw before, we observe this aberrant formation, these distinct monocyte macrophage trajectories, and this overall excessive inflammation coming from the myeloid progeny. Now, with the time that I have left, yes, I'm good with time, I will explain the third project that it was. The idea of this project was, okay, it was kind of knowing the field anyway that inflammation, infection favors TET2-derived clonal hematopoiesis. So we wanted to do a project that the aim was to find an environmental situation that favors not pre-malignant clones. So for that, we team up with the group of Andreas Stram in Heidelberg to study the impact of erythropoietic stress. In this case, they provide this super cool cohort of individuals that they have been able to donate blood through 40 years of time, since their 20s until their 60s, more than 120 blood donations. This is an incredible erythropoietic and sustained stress over time. This population is actually, you have to consider that we are analyzing here quite a unique and healthy population, because it's quite rare that individuals that they are 60 or 70 years of age, they are still able to, because they match the criteria to still donate blood. And we analyze the clonal hematopoiesis in this cohort. As you can see here, there is no significant increase of clonal hematopoiesis in this frequent donor cohort, not the overall incidence and not clonal hematopoiesis associated with DNMT3A or TET2. But as we dig more into the analysis, we realize that, and this is focusing on DNMT3A, we realize that the type and profile of mutations in the DNMT3A gene was different between the two cohorts. And the mutations in the DNMT3A in the frequent donor cohort were mutations that are predicted to be destabilizing mutations for the DNMT3A protein. So we wanted to recreate these mutations in our system. As I showed you before, by CRISPR, we introduce in cord blood or bone marrow hematopoietic stem cells this mutation. And in this case, we introduce EPO as a stress, as an environmental condition, as we thought that the erythropoietic stress, and because in which every blood donation there is a peak in erythropoietin levels, this might be the environmental condition that can select for these DNMT3A variants. And we compare the dynamic of these clones with inflammatory stresses, as I showed you before. So these are three different DNMT3A variants that we specifically found in the frequent donor cohort, and we observe a significant expansion of these variants upon EPO-rich environments. And we actually compare the dynamics of these particular clones with the DNMT3A clones that are associated with leukemia, as you know, they are 8A2 clones that are reported, and we also validate this idea that these clones expand in inflammatory environments. In this case, upon interferon gamma exposure, we have a significant expansion of R8A2 clones, while these variants, these DNMT3A variants that we found in the frequent donor core, they don't expand in inflammatory environments. They do expand significantly upon these EPO-rich environments. So this is an interesting difference and discrepancy in the clonal dynamics between two DNMT3A variants. And actually, this is the case also. This was a bit of a reviewer question. If you mix them and you have both at the same time in the same system, the frequent donor variant and the pre-leukemic variant, how they behave. And it's also the same. So basically, the frequent donor variant, it will be favored in EPO-rich environment, while the pre-leukemic variant will be favored and outgrowth in inflammatory environments. So this competition and the different dynamic of these clones also happen when they are together. So then we also perform this tapestry analysis to understand the incidence of these DNMT3A variants within the hematopoietic tree. As you can see here, for the R8A2, the pre-leukemic variant, we observe that is associated with a myeloid expansion, as we were expecting, but our blood DNMT3 variant doesn't cause any significant lineage bias. And this was in a steady state in these individuals after donating blood. Now, if we analyze this with our humanized mouse model in a more acute phase of the erythropoietic stress after a regimen of bleeding and EPO injections, what we observe is that, again, the pre-leukemic variant is associated with a myeloid expansion, while the frequent donor variant actually doesn't cause any lineage bias if something upon this acute phase of the erythropoietic stress is actually enriched or more present in the erythropoietic lineage, in the erythroid lineage. So the conclusions of this project, that again, it was a very nice collaboration with Andreas Strand group. It was the frequent donor, frequent blood donation does not increase clonal hematopoiesis, which was also something very important for us during the revision process to really be confident that, of course, blood donation is totally safe regarding incidence of clonal hematopoiesis. And there is this specific association of EPO-responsive DNMT3A variants and the distinct growth dynamic between these variants and the pre-leukemic DNMT3A variants. And this, for my future, is a bit of a project that makes me think in this future direction and projects to try to find and characterize environmental conditions that could select for non-malignant clones. Because at the end, the way I see it is that as we age and we encounter multiple types of environmental stresses, our polyclonal complexity is going to be reduced. This is clear. And if the malignant clones or these pre-malignant clones are going to outgrow, it's good to find some alternative that could counteract this pre-malignant clone expansion. And finally, another future direction that I would like also to discuss with you maybe later is this idea of how the influence of clonal hematopoiesis in tissue homeostasis, as you know, as we have seen at the beginning, some of this, we touch a bit on that on the cell stem cell paper, how the clonal hematopoiesis is going to infiltrate other tissues and, for example, can shape the tumor microenvironment in the lung, for example, or the immune response upon certain infections and things like this. So I will be happy to discuss about it. And with that, I will finish just by acknowledging Dominic's lab, her support, and all the members of the team, and also especially the collaborators as well, because this was, if something, thanks to collaborators as well, Benny for the collaboration with the neutrophil and the net quantification, also clinicians from the UK, and especially Andrea Stram for the DNMT3 project, and Elisa Laurenti, Caetano for these other projects that I'm starting independently. And of course, also Charlie Swanton and Uriol Peach we are helping them in characterizing the influence of chips in the context of lung cancer. And thank you all for your attention.